It's 11:45. So I'm Adrian Mander, so I'm going to introduce the next session on novel trial designs. And just sort of background information, I've been working in trials for 25 years. And when I started in trials research, most trials were two-arm trials, and there was not much complexity about them. And some of these novel designs that we're going to be talking about in this session were just ideas by methodologists. And these days, and possibly because in the olden days, trials had, we're still looking for new drugs in new areas, and we found a lot of drugs, we found a lot of interventions, and now we're trying to make them more and more efficient because also costs are going up, and novel designs is possible solution to this. So novel designs are essentially something beyond the two-arm trial. Often we look at information during the trial as we go through it and make decisions about it. A classic example, which is going now, the recovery trial, the COVID trial. And during that trial, that's an extremely novel design. It's allowing treatments to come in and out. It's very flexible. And it means that you can answer lots of things in sort of more real time. So they had one treatment arm, hydroxychloroquine, which was dropped during that trial, and for not being that effective. But equally, they've identified treatments that do work, like prednisolone or steroids. So you can see that people want to do something more flexibly and give us a bit more efficiency back to the trials. And they're trying to answer lots of different questions within a single trial, whereas before we had single trials, single questions, very simple things. And even trying to combine things faster from early. I've seen phase 1 trials where you're looking at whether a treatment is toxic to healthy volunteers or diseased participants, and they were very fixed doses, and you escalate in a certain way. And now you're finding things, changing doses rapidly and then moving on to the next stage very quickly. So they have these sort of seamless trials where they run one into the next. And a lot of this innovation was driven by oncology, and possibly because in the past there were few therapies, and now there are more and more therapies. And we are looking now at trying to measure things in the blood to say whether this treatment might work better than another treatment. So, but this innovation from oncology is now being driven across all different therapeutic areas. And the 3 talks we have today are going to cover different areas, one in oncology, one in type 1 diabetes, and the other in various endpoints. So we have 4 speakers today, and the first one's going to be Charlotte. And she's going to start talking about master protocols and platform trials in type 1 diabetes. So I'm going to pass over to you, Charlotte. Charlotte, we can't actually hear you, or I can't hear you. You're not on mute, I can see that. No, can't hear you now, unfortunately. I'm wondering if it might be better while you're having technical problems, maybe you need a reboot or something, and that maybe we should go to the next speaker. Would that be reasonable? Oh, I still can't hear you, Charlotte, unfortunately. Maybe we should. Sorry, maybe Charlotte. Maybe you need to reboot your machine, and then if perhaps we go to Ian. With that, is that okay, Ian?
Yeah. Can you hear me?
Now I can hear you loud and clear.
That's
So
a good start.
he's going to talk about AML trials. So over to you when you share your screen.
Come on. Okay. Adrian, can you hear and see me properly?
I can. Everything's great. Thank you. Over to you.
Well, so far. Okay, so sorry to push in, Charlotte, but hopefully this will help you out a little bit. So I'm going to talk about novel clinical trial design. As Adrian said, trial design has moved on quite a lot in the last 15 or 20 years. I'm going to talk about it in the context of rare disease. And this is more of a case study, really, about a disease called acute myeloid leukemia, or AML. I'll give you some numbers as background. This is the most boring slide, so I'll get it out of the way first. So, number of cancer diagnoses in the UK is reasonably stable at around about 350,000, which is a pretty depressing number. I think we can all agree. The leukemia diagnoses within that is about 10,000, and the acute myeloid leukemia diagnoses, quite a small number, i.e. it being a rare disease, of about 3,000 per year. So you've got the same chance of being diagnosed with AML every year as requiring a kidney transplant, being killed in a road accident, injured by a firework, or, to put a positive slant on it, becoming a professional athlete, which wouldn't usually be a surprise to people who become a professional athlete, I wouldn't have thought. So AML, as you can see from those numbers, accounts for less than 1% of cancer diagnoses, but more than 2% of cancer deaths every year. So it is, even within cancer, it's a nasty one. Okay, so looking at the age-gender split, you can see that this is predominantly a disease of the elderly. I would like to say this is a CIUK graph, so this color coding of pink for female and blue for male is straight out of the 1970s, but it is from CIUK, so that's not me. We tend to split the AML population into over 60s and under 60s. That's a bit of a traditional sort of throwback that we're trying to break down. Under 60s will all receive intensive chemotherapy. Over 60s, more and more, with an aging population and a fitter aging population, elderly patients will receive intensive chemotherapy, but there are a large number who are too frail and receive non-intensive chemotherapy. So we've got different approaches for those two different populations. So what does a pathway look like for intensive treatment? Well, a patient would ordinarily turn up to a regional health centre. They would be punctured for a bone marrow sample and then start receiving intensive chemotherapy. So this is a blood cancer. We can't enact any surgery, and radiotherapy would be ineffective because there's nothing to really target. So in AML, our only weapon is drugs, really. So the intensive chemotherapy would start. Obviously, it's an unpleasant experience. It would be followed up by another bone marrow sample to see what sort of response we've got from the patient. So by this point, we would have a fair idea of the patient's genetic profile as well. A portion of this more recent sample would be analysed locally, but also, as Adrian mentioned, the scientific sort of background to oncology. There are active research groups around the country and around the world who are always looking for more sensitive markers, more sensitive ways of analysing response to treatment. So we try and get involved in that and support those scientists. All that initial phase would traditionally happen in a regional centre. So I'm sat here in Rhondda Cynon Taf. if I was diagnosed with AML, I would probably receive this phase of treatment in UHW in Cardiff. There's always a discussion about number of courses that's best for any patient to receive. That's dependent on their response and their age. So there's debate about that. At the point where we've assessed the patient's response to their initial treatment, we might want to give them some more treatment on a longer term basis, maintenance treatment. If they're not doing so well, we would need to up the ante, so to speak, to get that patient ready for a bone marrow transplant, which is particularly unpleasant and particularly expensive. If they were doing well, they might be referred on to longer term follow-up, which would be, let's say, 6 or 3 monthly bone marrow sampling. And any of that might happen in a different hospital. I'm sat a mile away from the Royal Glamorgan, so a longer term follow-up may happen there. If you're situated on the other side of Newport, your transplant might happen in Cardiff, it might happen in Bristol. So it's a fluid population, as in many diseases. So looking at the constituent parts, what are we trying to actually achieve with our clinical trials and what we're trying to improve on? So intensive treatment is reasonably effective now. So we've got a reasonable standard of care. So if a pharmaceutical company derive a novel treatment, then the comparison with standard of care that they would need to undertake would require many thousands of patients potentially. That would be a 5 to 10 year undertaking, and if the pharmaceutical company were to undertake that on their own, they'd be looking at a 10 to 20 million pound clinical trial. With regard to number of courses, what we've achieved as a group over the past 20 to 30 years is to significantly reduce the number of chemotherapy courses that an average patient would receive. So back in the 1970s and 80s, patients would potentially just receive courses of chemotherapy until their disease either progressed or the doctor felt that they were stable enough. That was never supported by any robust evidence. So we want to reduce the amount of treatment that patients receive without significantly decreasing their outcome or depleting their outcome. In order to sort of undertake that sort of investigation, we need to know what treatments the patients have received. You probably need less patients to do that sort of analysis, but equally long-term follow-up as you would need in the intensive treatment. And any scientific lab-based elements, the laboratory data needs to be compared to the clinical outcome data, of course, for it to be meaningful. Most of these lab groups are looking for particularly sensitive markers, but that is reasonably meaningless unless it tells us something about the patient's disease. Lab-based work, as we probably all know, is pretty expensive from a staff, resource, setup point of view, but also if that marker is not specific to a particular drug, then you might find it difficult to get drug companies to support any particular investigation. Okay, so how do we try and improve things in all those scenarios without taking forever? So what we've introduced traditionally, not introduced, it's been part of the sort of makeup of the AML treatment for a long time is this platform trial design, which Adrian touched on in his introduction. So what we have is a protocol which is made available to all the hospitals in the UK. Additionally, beyond the UK, across Europe and in Australia, New Zealand, traditionally have supported our trial protocols. So here we have a question in relation to induction chemotherapy. Standard of treatment is questionable, but we can always add a novel treatment to it. Potentially 500 to 1,000 patients would go into each arm here. There'll be an assessment after induction chemotherapy for each patient. We've also got a question there to consider the right number of courses for these patients, and also a laboratory-based question, all within the same protocol. That particular question is more about whether laboratory interventions actually help the outcome of the patient, which is quite a fundamental question to be asking on behalf of the patients. So you've got a patient coming in here, randomized, they might go on to this induction chemotherapy, they have their post-chemotherapy analysis, they may be assigned to this particular treatment pathway, and then if they give their consent, they would enter that number of courses investigation. Similarly, another patient may come along, they have a different induction chemotherapy, another assessment as per the other patient, but unless they consent, they wouldn't necessarily travel through the protocol. So it's a flexible approach, which is based on whether the patient feels they can support these different questions, and also with the variety of healthcare settings that we make this protocol available to, different hospitals may want to support different parts of the protocol. And that's important because all of the data needs to reflect the real healthcare setting. If we just open this in large teaching hospitals, it might give us a slightly different answer to our question. So that's how we've approached the intensive treatment pathway. In patients where frail elderly patients who receive non-intensive treatment, it's a slightly different background. So this graph hasn't come up very well, unfortunately, but this is the improvement in outcome in patients who do receive and are suitable for intensive chemotherapy. So the black line down the bottom of this graph is denotes outcome in the 1970s, and the light blue is the improvement in stages up to probably 15 years ago. So you can see that survival at 5 years back in 1970 was down around 10%. 15 years ago, it was around about 50%, which is a considerable improvement, of course. I could say that probably in 2020, that's closer to 60–65%. Unfortunately, in the patients who receive non-intensive treatment, there hasn't been a significant improvement in outcome during that time. What outcome improvement that we have seen is probably related to improvements in supportive care and just general improvements in healthcare delivery, rather than any improvements in the treatment that we give them. But if we consider the sort of age spread that we looked at in the slide earlier in the slide set, a large proportion of patients who are newly diagnosed with AML are elderly and frail. So this is a large population that we're not serving very well and haven't improved for a long time. Given that, there has always been enthusiasm in the pharma world to try and get some drugs into these patients and get their drugs analysed in this patient population. This is just a small number. What's that? 14, 15 of the novel agents that have been identified probably in the last 10 to 15 years as being possible improvements in this scenario. So, as Adrian mentioned in his introduction, a typical trial going back 10 or 15, 20 years would be a two-arm trial comparing standard treatment to whatever you feel is the next great thing. In this particular scenario, we would probably only need 100 patients to assess outcome in these elderly frail patients. We might put 50 in each arm and just have a quick look to see how many are in remission. With the extremely poor outcome in this group of patients, we don't really want to be improving outcome by 1 or 2%. We really want to be doubling the amount of patients who are alive at 5 years. Otherwise, we feel there's probably not the enthusiasm in healthcare systems to fund the drugs that we're looking at. So patients who get into remission will survive better. We can have a look at the percentage of patients in remission, but obviously we would still continue to look at their survival. So this sort of trial will, maybe I'm being optimistic, the trialists among us may may see 1 year setup as being an optimistic time frame in this time period, but only a year of recruitment will be required for putting 100 patients in, and sadly we would only need a year of follow-up to gain an answer with regard to survival in these patients. So if you consider that 3-year time period for 14 to 15 new drugs, putting them in a linear progression, that would take 40 to 45 years to answer any questions about those drugs. So what can we do to improve it? Well, we could potentially just leave that trial open, assess drug A. If it's good, the drug company might go away and conduct a bigger trial themselves. If it's not good, we would report on that. And then we could just replace drug A with drug B. We'd save our setup times. We could recruit while we're following up the patients who've received drug A. So is that what we do? Well, no, we can't do that, because that would still take 20 to 25 years to assess all the drugs that are available. So what we do is we throw them all into one big protocol, and we assess everything against standard treatment at the same time. So here we've got a patient entering the trial. They may be allocated to this particular novel agent. Now, elderly frail patients would receive the same treatment sequentially and repetitively. So if they were assigned to standard treatment, they would receive X number of courses of standard treatment until their disease progressed. Similarly, if they receive novel treatment, they would receive that on a repetitive basis. So as we analyse each drug, we may feel that it's not showing any promise, so we would drop that from the protocol and add in another drug. So over the course of this trial was set up in 2010. Over the course of the last 10 years, we have pretty much moved from the options that were available at the trial outset, above standard treatment here, and added in all these new drugs. So the assessments have been pretty much on an annual basis. Obviously, with a number of novel drugs to put into a protocol and to convince hospitals that this is the best way to treat these patients, then we need additional setup time. Longer recruitment time, obviously, because we're assessing 8, 9, 10 novel agents rather than 1 or 2, but that recruitment time can be sequential rather than the inefficient design of the 2-arm trial that we mentioned earlier. And then follow-up is ongoing, because every time you drop an arm, you continue to follow up those patients, but you add a new arm in. So you are reporting on each arm of the trial as a new arm is added in. So like I said, this trial was set up originally in 2010. We now have about 30 patients left out of 1,100. And these are the only 2 options left available. So it has been a successful program of development, and it's been with me so long now it feels like a member of the family. So what else can we do? Because that's all very well, but as Adrian says, this is a reasonably common approach now, or not a common approach, but is being taken into other disease areas. Well, if we strip back the patient pathway, what aren't we doing? Well, I think the referral process has not been investigated fully for these patients. Traditionally, a patient will turn up to a GP feeling fatigue, possibly bruising that won't go away, maybe a bit of nausea, maybe a bit of vomiting, very vague symptoms. GP won't necessarily have any experience of AML. If a GP's probably particularly unlucky, they may see 2 or 3 AML patients in their career. So it's not something that's going to be at the top of their, not something that they would have been exposed to. So is this referral pathway working as well as it could? There were some talks earlier about inclusivity. Is there a socioeconomic element to how that referral pathway works? So if a patient's in a rural community in Brecon, are they getting to hospital to start this intensive treatment at the same rate as somebody in Chelsea or the centre of Manchester? You would assume not, but we don't even investigate this at the moment, which I think can be corrected. Chemotherapy works in this scenario to clear out the patient's disease. It also effectively clears out their immune system. So obviously these patients are very prone to infection, fungal or bacterial. And this is something that we don't particularly investigate in our trials. CTR have got a very active, very successful infections group. And that's something that we can include in future trials in these patients for their benefit. Nutrition is a big issue with any patients who receive chemotherapy. Patients who can sustain a suitable nutritional regime do significantly better than those who can't, but that's a question that we don't include in our trials at the moment. And similarly, exercise has been difficult to measure probably until about 5 to 10 years ago, but the access to technology like Fitbits should be able to inform us of the quality of life and exercise regimes of different patients on different treatments. It could well be that patients think that is a critical part of assessing chemotherapy. We don't know that because sadly we don't measure this at the moment. So that's a sort of a very quick run through of how we use novel design in AML to try and answer a number of questions at the same time in a platform design, and also to test a number of different drugs at the same time in what we would call a multi-arm trial. I just want a quick acknowledgement to the people in Cardiff who've helped support these trials over the years, and nationally we obviously need some leadership, and locally we need people to keep us in line to make sure we're doing things properly. But significantly, we've put tens of thousands of patients into these trials over the years, and without them I wouldn't be able to metaphorically wave my arms around in front of some slides and report this back to you. So hats off to them.
Thank you very much, Ian. And yeah, it's amazing how complicated some of these designs are, and I'm sure people have got lots of questions about those designs, so keep the Q&A coming. We're gonna try and calling Charlotte.
Fingers crossed.
We have sound.
Let me try, okay?
I can hear you. So
can you hear me now?
Yeah. So
okay.
Charlotte's going to talk about novel designs in type 1 diabetes and INNODIA. Over to you.
Can you still hear me? Thank you. Okay, sorry about the technical difficulties. It's a new laptop and clearly issues. I'd like to talk to you about master protocols and platform trials in type 1 diabetes, which are adaptive designs from the INNODIA consortium. And these were created in conjunction with myself and Professor Adrian Mander. So first of all, what is a platform trial? So a platform trial is a single trial to evaluate multiple treatments or treatment combinations and using the same protocol. So a platform trial contains many sub-trials. So the INNODIA consortium, for instance, includes the MELD-ATG trial and VERITID-1D, which I'll talk about in just a minute. It can be a perpetual trial, so have no end. And we consider these sub-trials, so again, MELD-ATG and VERITID-1D would be those trials, as independent trials. So we do not adjust for multiplicity. Now, let's say that we do decide to compare between these sub-trials, asking perhaps the question, which of these treatments is the most efficacious? Then in that case, we would have to control for multiplicity or for overall false positives. So it's recommended that these platform trials have a common control so that you're comparing treatments to concurrent controls which are matched in time. Any kind of indirect comparisons would have to be done using models. Now let's say that these sub-trials do not include a shared control data, then we can use the natural history data in conjunction with placebo data to enable such comparisons. And we often use Bayesian methods, which I know can be a scary word, to measure how closely the placebo data are to the natural history data. And ideally, our sub-trial population is a random sample of our natural history data. So why do we use Bayesian statistics in a lot of sort of these adaptive designs? So I've created a graph for you on the left-hand side of your screen, which looks at the relationship between the Bayesian prior, that's your red curve, the posterior, the blue curve, as well as data, the black curve. What I've plotted for you on the x-axis is treatment effect of, let's say, a potential drug or intervention. And on the y-axis, I'm showing you the frequency of seeing that treatment effect within the prior curve, the red curve, the data curve, the black curve, or the posterior curve, the blue curve. So Bayesian statistics uses external evidence from past and concurrent studies, databases, or even expert opinion, which is expressed as a prior probability, or your red curve. And traditional statistics tends to just use evidence from new data, that is your black curve. And often that's done in a model. However, with Bayesian statistics, we combine these two sources of information to give the posterior probability in blue. So why this is really useful is that in the cases of small sample sizes, where let's say we don't have a lot of new information to add to our model, or something where we have longitudinal data and we haven't really collected enough information, but we have a lot of prior information, we can still detect an effect with a posterior probability strongly following the prior probability. So in that case, your blue curve would closely mirror your red curve. However, if we don't have a lot of prior information, but we have a large sample of new data, then the posterior probability will follow the likelihood or new data more strongly. So that's sort of what you're seeing here on the graph, where you're seeing that the blue curve is mirroring your black curve. So the INNODIA consortium is very large. It's made up of 26 academic institutions all over Europe, 2 patient organizations, 4 pharmaceutical partners, as well as several CROs now. So Cardiff University is part of it. As you can see, it's listed below the academic partners. And the idea are to develop a clinical European infrastructure to recruit a deep phenotype and biosample type 1 diabetes subjects at diagnosis and their at-risk relatives, which allows also the possibility of not only treatment studies, but as well as prevention studies. We want to establish a tight collaborative network of researchers, and also to establish a unique integrated database of innovative research and data to support the search for a cure, but as well as to advance the development and application of novel methodologies and to conceive of innovative clinical trial design. This is actually being done by us here at Cardiff University. So 2 of the sub-trials in the INNODIA consortium which are planned are adaptive trials. The first is VERITID-1D, which is a 2-arm trial, which is in adults, and it uses a model on the repeatedly measured outcome of C-peptide. Now, C-peptide is a measure of disease status or disease burden for type 1 diabetes, and it's measured in the blood. For VERITID-1D, we plan to stop for futility, and 45 out of the total 120 participants have been recruited. The second sub-trial is also adaptive, is MELD-ATG, the dose-ranging trial. So for the first few patients recruited, age is restricted to those patients that are 12 to 25 years of age, but provided that we don't see any safety concerns in the first 10 participants to receive the ATG drug dose, we're going to progress to all stages, which in this case is also in very young children, so ages 5 to 25 years old, which goes back to sort of the issue of inclusivity, which we talked about previously. So we then use a Bayesian model in our interim analyses to predict which intermediate doses should be used. So first to discuss VERITID-1D. So patients will then first go through an enrollment and consent visit, and then they will have their INNODIA baseline visits and screening. If they're not found to be eligible for VERITID-1D, they will be asked to join the INNODIA natural history study, which goes back to the first slide, which we can use for comparisons within other sub-trials. If they are found to be eligible, they will be randomized 2-to-1 between the treatment arm, which is verapamil, against placebo. During that time, there will be an interim analysis, and these participants will be followed up for 12 months, at which point the study will end. And we need 120 subjects to be recruited, so 40 participants in our placebo or control arm, and 80 participants in our verapamil or experimental arm, detect a change of 0.18 in our C-peptide measure, which is again measured in blood as a marker of disease burden of type 1 diabetes. So to reiterate, C-peptide is repeatedly measured at baseline, 3, 6, 9, and 12 months, and again is measured in blood to show disease status. Our primary analysis is planned for 12 months, but we have an interim or futility analysis planned at 10 months. Recruitment is predicted to be around 5 per month, assuming a uniform recruitment pattern. But the idea is to fit a mixed effects model to estimate the treatment effect at 6 months and to stop for futility when 45 out of the total 120 participants have been recruited. So this gives us a 30% chance of stopping if there is no true treatment effect with verapamil. Now let's say the difference with C-peptide level is 18% when we're comparing verapamil to placebo. Then we have an 87% chance of detecting that treatment effect. The futility of interim analysis, including it, does reduce our power slightly. And our power, again, is the likelihood of the study to detect a treatment effect if there is one. However, the potential gain of including this interim or futility analysis is that we may need 23 fewer participants recruited to the trial. And indeed, if there is no treatment effect, we would need only 97 patients to actually show that. So the second adaptive trial to discuss in the INNODIA consortium is the MELD ATG trial. So for this one, we're recruiting a total of 114 participants who are going to be randomized into 7 cohorts sequentially. So cohort 1 will consist of 30 participants, which will be randomized equally into the 5 treatment options. These are placebo, it's 2.5 ATG, which is our highest dose of ATG, as well as the middle doses of ATG, which are 0.1 ATG, 0.5 ATG, and 1.5 ATG. Now, after cohort 1 has been fully recruited and their data has been gathered, and before recruitment to cohort 2 can begin, but again, recruitment will not be halted here, the dose determining committee comes together and looks at all the data for cohort 1 to determine which 2 best middle doses should be carried forward to cohort 2. Now, for cohort 2, 12 participants are randomized equally into the placebo arm, the higher dose of ATG, which is again 2.5, as well as 2 middle doses. The process continues, and before recruitment to cohort 3 can begin, the dose determining committee looks at all the accumulated data for cohort 1, as well as cohort 2, to determine the best 2 middle doses to carry forward to cohort 3. And so this goes on for cohorts 4 through 7, except for this one, we only are carrying over 1 middle dose. And we have 15 participants, which are randomized equally into our placebo arm, our 2.5 arm, as well as that middle dose. Now let's say that all the clinical information, safety information, as well as our Bayesian model doesn't find an appropriate middle dose to carry forward. Those patients will be allocated the 2.5 ATG dose because to improve power. So then those participants are followed up for 12 months, 1 to 4 weeks, as well as 3, 6, and 12 months post-treatment. Our end of trial analysis is looking at the effect of 2.5 ATG on C-peptide at 12 months as compared to placebo. And if we find that to be statistically significant, we will look to find the minimally effective dose of ATG. So again, those would be our middle doses. So again, 0.1 ATG, 0.5 ATG, or 1.5 ATG on C-peptide at 12 months as compared to placebo. So then participants the trial will be over, and they will be offered to join the INNODIA longitudinal study. So again, for this trial, C-peptide is also measured at baseline, 3, 6, 9, and 12 months, showing again this master protocol. And we have 6 interim analyses after the first 6 cohorts to determine which dose in the case of cohorts 4 through 7 will be administered to each subsequent cohort of participants using all available data. So again, the dose determining committee is convened to determine the next best middle doses to be applied to the next cohort. And in order to do that, they're going to look at not only CD4 and CD8 T cell outcome at 1, 2, 4, and 12 weeks. And if we know that if these levels don't change, and that dose potentially is inactive, so we will remove that dose for our future Bayesian prediction interim analyses. We will also bring together the clinical opinion of members of our dose determining committee, as well as safety outcome information from our previous cohorts data. Finally, we will use recommendations from a Bayesian repeated measures model. And this model looks at the predictive probability that each dose of the set will differ in means from the placebo group. So let's say that the study has been going on for a little bit, and we're about to select our final dose to be carried over for cohort 7. So the dose determining committee will come together, and one of the pieces of data that I will show them will be the mean difference in C-peptide with placebo. And obviously we want a difference that is greater than 0. So what I've plotted for you here in the red dotted line is sort of 0, which would mean no difference with placebo. And to the right-hand side, in the darker shading for each of these plots, you see that there's a greater difference with placebo than 0. And it shows you the probability of being above 0 for mean difference with placebo. The key thing to note here is that this data is obviously not real data, it's simulated data because the trial is only starting next month, but I've run these simulations over 1,000 times. And we see that for the 0.1 dose through these simulations, we have an 83% probability of being above 0 for mean difference with placebo. For the 0.5 dose, there was more variability in this dose, and we have a 70% probability of being above 0. For the 1.5 dose, it was an 81% probability, and the 2.5 dose, a very high probability, or 98% probability. And you can see that the curve for the 2.5 dose is mostly shaded in dark green. So to conclude, master protocols are difficult to write and they're complex, but they do lead to operational efficiencies. They're standardized, they have common quality control, they have one database across multiple studies, and the nice thing is that each trial can add additional measurements to the existing master protocol skeleton. The shared controls and the natural history data, and again, this would come from patients not eligible for that particular sub-study, should lead to statistical efficiencies and a better understanding of results between trials. So that's it for me. I will pass over to Phil, our next presenter. So please hold your questions to the end of this session. Thank you.
Thank you, Charlotte, for a very detailed explanation of the complicated world that is type 1 diabetes and INNODIA. So as Charlotte said, Phil's up next, and he's going to be talking about biomarker-driven designs. And whenever you're ready, Phil, share your screen.
Thanks, Adrian. Can you see my full screen?
Yes, we can. It's not on display, you know. You can see your full screen. You haven't got it on presenter mode. So if you click that little button, then you're ready to go.
Here we go.
Great.
Sorry about the little technical difficulties. Good afternoon, everyone. I'm going to talk about basket trial designs in the context of targeted therapies. So what do I mean by targeted therapies? Targeted therapies are therapies that are tailored to individual patients, which is what we call personalized medicine or precision medicine or stratified medicine. So the idea of this approach is actually quite simple and not particularly new. The idea is simply that patients get the treatment that works best for them, or that we think works best for them. So this approach acknowledges the simple truth that we as humans, as patients, are all different, are diverse, and sometimes in very obvious ways. So here the chap on the left-hand side is a very friendly chap. He offers the tiny chap on the right-hand side one of his pills, but it's quite obvious that the dose is going to be way too high for him. But we are also different in ways that might not be immediately visible, for example, on a genetic level, on a molecular level. So what personalized medicine does is it moves away from a one-size-fits-all approach of medicine and tries to introduce targeted therapies that achieve the best outcomes for individual patients, which means the same disease might be different in different people and therefore might require different approaches to treatment for different people. So at the end of the day, we're treating the patient rather than the disease. And here's a quite simplistic illustration of this principle. So in the traditional non-personalized medicine approach, we have this diverse population of patients, some of them red, some of them purple, some of them blue here in this illustration. We have one type of therapy, and it happens to have a positive effect on a subpopulation, in this case, the blue guys, whereas it doesn't have any effect whatsoever on the red people, who seem to be the majority of the population. And even worse, it has a negative effect on some people. That's clearly not what we want. So therefore, personalized approach to medicine introduces another step, and this additional step is a screening test. So before we decide which treatment to recommend for a specific individual, what we do is screen them, which might be a biomarker test, a blood test, gene test, and then we stratify our treatment options based on the results of that screening test. So in this illustration, the red people get the red syringe, the blue people get the blue syringe, and the purple people get the purple syringe. And then we hope that everyone has a positive outcome. At least that's the dream. But of course, it requires a screening test in combination with different treatment options for different types of people or subpopulations. And as we develop these kinds of treatments and screening tests, it's pretty obvious that the classical two-arm randomized control trial is not good enough to do that. So what we need is a kind of personalized way of running clinical trials, so that trials adapt to the new way of practicing medicine at the end of the day. And that's where basket trials come in. So basket trials are one of those novel types of designs that can be used to develop targeted or personalized therapies. So when aiming to improve trial designs for those kinds of therapies, I kind of like comparing basket trials to basketball, not only because it involves a basket, obviously, but I think personally, basketball is a wonderful sport, but it's clearly not the right sport for me. I'm very short, I have terrible aim, there's no way I can reach the hoop or dunk the ball into the hoop. I'm also quite lazy, so I don't like to run. And so it's the perfect sport, but not for everyone. And instead of this one-size-fits-all approach, I think people like me would enjoy basketball, at least playing basketball, much more if it looked like that. And that's essentially what we're trying to do with these therapies and also with these trials that are targeted. So I'd be very happy to dunk the basketball into that very low-hanging hoop. And this is what some of those novel designs are about, having the right treatment or the right basketball hoop for the right person. So let's have a quick look at traditional trials in comparison to basket trials and another type of novel designs called umbrella trials. So in the traditional two-arm, or in this case, three-arm clinical trial, we have several treatment options, in this case, a blue pill, an orange or yellow pill, and a green pill. And we want to know which one is best. But this approach, which recruits from the whole population, which is diverse, is not much more than a shotgun approach, because people randomly end up in treatment arms, and just so happens that the blue pill works best for the blue people, the yellow pill works best for the yellow people, and the green pill works best for the green people. And in each arm, we end up with lucky few that get the right treatment and lots of people who don't. So what we want for precision medicine is a better approach. This is an example in lung cancer. And as Adrian mentioned earlier, most of these novel designs have been pioneered in oncology and same here for umbrella and basket designs. So this is a very simple example of a trial, of an umbrella trial in lung cancer. Lung cancer can be very different in different people. So the genetic mutation that causes the lung cancer might be different in different people, or therefore the cellular target that is targeted by the drug will also have to be different. And therefore, we cannot just have one treatment for lung cancer, but there are so many different subtypes of lung cancer, and it will be more effective to treat each individual patient based on what subtype they are. That's the idea of, and therefore more people get the right treatment that's right for them. That's the idea of precision medicine or personalized medicine. And basket designs, in which I will focus in the remainder of the talk, are another way, albeit a different one, of doing exactly that. So we have a specific genetic or molecular target, and in a basket design, the treatment is the same for everyone, but we have different types of cancer in this example. So we have people with ovarian cancer, we have people with gastric cancer, we have people with lung cancer. But what is common to all these cancers is that they have a similar or the same genetic mutation, underlying genetic mutation or molecular target, which means at the end of the day for chemotherapy drug, it doesn't really matter where the tumor sits, as long as the molecular target is similar. So what we're doing in a basket trial is we include baskets with different subtypes of disease, lung cancer, gastric cancer, ovarian cancer, and so on and so forth, all in the same trial, which is very efficient because, as Charlotte just mentioned a few minutes ago, we can have one overarching master protocol, and we're essentially running 5 sub-trials in one. And I'm going to show you an example, not in cancer, of a basket trial that we're in the process of setting up at the moment. This trial is called DocTIS, Decision on Optimal Combinatorial Therapies in IMIDs. I'll tell you what IMIDs are in a minute, using systems approach. So this European consortium project funded by the EU Horizon 2020 program, and it includes a work package for clinical trial that will start in early 2022. Chief investigator of that is Professor Ernst Join, School of Medicine here in Cardiff, and he will be running this trial in collaboration with Dave Gillespie and Ian Thomas, who just presented earlier in this session, and myself, and I'm sure other colleagues from CTR will join the team as we move along. This trial will use a basket design. So what's the aim of DocTIS? The idea of this project is that we find a combinatorial therapy, which means a combination of existing drugs, which is to be determined at one of the other work packages by one of the other European partners. And we'll also find a biomarker profile or biomarker signature, which means we can apply a simple test, probably a blood test, and test whether a potential patient is likely to benefit from that therapy. So we will know by the time we start the trial which type of patient, which subgroup of patients will likely or more likely benefit from the therapy. And then we will trial this new therapy, this new combination of existing therapies in a variety of IMIDs, which are immune-mediated inflammatory disease. And you can see these diseases here. We have 6 baskets in this basket trial. So what one would traditionally do is run 6 separate trials. What we're doing here is we're running one trial, one basket trial across all those different conditions, which is obviously highly efficient because we can run them all in parallel using the same overarching master protocol. What will happen is will screen potential trial participants who are patients with one of these diseases using a biomarker or genetic test. And if they screen positive, which means if they have a good chance of benefiting from the combinatorial therapy that is yet to be identified, then we will include them in the trial. And this is a bit like playing basketball with this multi-hoop, which is, by the way, an actual thing. This is not Photoshop. So who will these participants be? They will have one of these 6 diseases. Conventional therapy will not work well for them, and alternatives are much needed, which is why we run this trial. And they will obviously have that positive biomarker profile. It's a multi-center and multinational study. We aim to recruit 80 patients across these 6 different IMIDs from at least 34 hospitals in 4 countries in Europe. The outcomes of the trial will be, so the primary outcome will be the percentage of participants who improve after 6 months of receiving this new combinatorial therapy. And what is important is that disease remission will be defined differently for the 6 diseases because they are different diseases, and that's something the basket design allows us to do. So we can use disease-specific criteria for remission or improvement. We also have secondary outcomes, which will be safety, disease activity, again based on disease-specific activity scores, and generic health-related quality of life score. So in terms of the design, overall we have this basket design, but within each IMID, within each disease, we will run an open label, which means patients will know what they get and their doctors will know what they get. Multi-center adaptive 2-stage single-arm study. So this is an illustration of this adaptive 2-stage single-arm study, which will use a design called Simon's design or a similar design. What we do is we start recruiting patients. It's called stage 1. If their remission rate or the percentage of people who show improvements is below a certain threshold, we stop recruiting patients with this disease because there's a clear lack of efficacy for this patient population. But if it's above a threshold, we continue recruiting, and at the end of the study, we either conclude that the combinatorial therapy is efficacious or not. So what this design allows us to do is to stop recruiting, that is to stop wasting resources and efforts on investigating this drug in patient groups, diseases where it clearly doesn't work, which again adds to the efficiency of the design. This is also called pruning. So we prune IMIDs for which the new therapy doesn't show sufficient response, and therefore focus on those where there's more promise. And another thing we can do at the end of the study is pooling the results so that we don't just analyze whether the new therapy has an effect within each disease, but also across each disease. So we can group them, or we can then draw overall conclusions or conclusions across multiple IMIDs. And we can do that for all 6 diseases within one trial. And that's pretty much the end of my talk. All I want to say, the next slide, is that this type of design is something we intend to use in more trials. This is just a backup slide in case I run out of content before I run out of time, but I don't think that this is happening. So DocTIS is not going to be the only basket trial in CTR for long. We've recently submitted a proposal, including this graph, which which is currently under review, so I can't go into any details here. The idea is that we have a new non-pharmacological treatment based on physical activity programs, and we hypothesize that this will help improve people with different neurological conditions, including muscle disease, peripheral nerve disease. So again, we have these different types of diseases, and they will all be included in the same trial. And within each group of diseases, we will have a randomized 2-arm trial, and then we can again prune baskets for which there is no sufficient promise. And at the end of the day, we can analyze each basket individually and together in a pooled way. And this is truly my final slide. Just want to acknowledge that DocTIS has received funding from the EU's Horizon 2020 program, and happy to answer any questions in the discussion session that follows. Thanks very much.
Great, thank you so much, Phil. It is interesting that the European Commission does fund a lot of these adaptive trials and complex novel designs, which is great. So our next speaker is Lucy, and she's going to take us through some of the innovations within quality.
Okay, can you hear me?
I can hear you.
You can hear me. Oh, good. That was easier than I thought. Okay, so hopefully you can see the slide. So yeah, thank you. I'm going to talk today about embedding qualitative research within trials and the work of the CTR Qualitative Research Group. So a little bit different to what we've been hearing about so far, perhaps. But within the CTR, we have a qualitative research group consisting of over 20 experienced qualitative researchers. Some, but not all of them, are featured here in our gallery. I think it's been updated for a while, so apologies to those who aren't on here. CTR is in a lucky position in that it's one of the few trials units in the UK where we have a group of qualitative researchers firmly embedded within the trials unit. So our qualitative researchers have a real understanding of the practicalities of trials because they work on a day-to-day basis alongside trial managers and data managers. We work in interdisciplinary teams with health economists and statisticians to solve complex problems. So the members of our qualitative research group have a wide range of experiences and backgrounds in, for example, psychology, sociology, sociolinguistics. So we really do bring quite a sort of particular area of methodological expertise to the trials unit. We have a group of creative researchers who adopt novel methods and use them in different ways to answer really very real and applied research questions and who present their research in a meaningful way. So I'm going to talk to you about the different ways we can use qualitative research within and alongside trials. And we can do this at various time points, so before, during and after trials. And also I want to talk to you about some of the methodological innovations that members of the group are working on in terms of using visual methods. So hopefully I'll have time to zoom through all this. There's quite a lot to cover because I'm going to give you some examples from the trials and studies we've worked on. So I won't necessarily go into huge details and the results of each trial, but we'll sort of tell you about the methods that we've used. Okay, so qualitative research methods can be used to understand health and illness before a trial and before any work has been done to formally develop an intervention. So data collection methods such as narrative interviews, focus groups, observations can be used to help understand how and why there's a particular phenomenon from the viewpoint of the individuals concerned, so the healthcare professionals or the patients themselves. And one recent example where we've done this is in the placement trial, where qualitative research was carried out alongside a randomized controlled trial pilot study, which looked at the feasibility of recruitment of patients into a trial using perineural local anaesthetic catheter after major lower limb amputation. So we carried out longitudinal qualitative interviews with patients, both as soon as was ethically possible after the amputation, and in terms of acceptability to when the patients felt it was appropriate, and also carried out interviews at a later point, again, when patients had returned home. So these interviews were to explore experiences of amputation and pain control, and the interview questions were very flexible to allow the patient to guide the conversation in the direction and to focus on the topics they thought were most important. So what did we find out? One of the things we found out was that patients, when they talked about coping and pain, tended to compare themselves with other patients in the hospital and on the ward. And so this element of competition is perhaps something that could be used in designing a future intervention. So we kind of think that that can help us understand and help us develop interventions in the future, possibly through stimulating friendly competition in group rehabilitation. But also, one of the other important aspects of carrying out this qualitative research was to understand more about the design of the trial itself, to then help us improve the design of a full trial in the future. So interviews with healthcare professionals showed, for example, that it was difficult to get the post-operative pain scores that the trial was hoping to get, particularly out of hours. So it's possible that a reduction in the amount of data collected post-operatively would increase feasibility. Also, both patients and the healthcare professionals reported that using more than one pain scale was confusing and often might give conflicting scores. So again, this can be taken on board in the design of future trials. We also found, very importantly, that in talking to patients, although they reported and had told us that they were happy with the trial processes and found them acceptable, their understanding of the trial at the time of the interview, they didn't necessarily demonstrate a good understanding of the trial. So while this may be due to the fact that patients could be struggling to recall information that was given to them some time ago, it also highlights the need for healthcare professionals to check patient consent, patient understanding at the time of consent. So again, this is something that can be built into future trial design with this population group. Okay, so moving on, once we've understood more about a problem or a phenomenon, we can use qualitative research methods to help develop an intervention. So qualitative methods can allow close collaboration between researchers and participants to help design interventions that respond to the particular needs of that patient group, but also make sure that the intervention doesn't just look good on paper, but also can translate into real life settings as well. Increasingly, there's been a call for studies to use the co-production approach, where researchers work closely with research participants to design an intervention together. So this is particularly with complex interventions. So a good example of this in a recent study was the Assist Frank study, where the chief investigator, Jamie White, worked with a qualitative researcher, Kim Smallman, and the group developed 2 drug prevention interventions, Assist Frank and Frank Friends. So the interventions were piloted in 1 school each, and they used a range of qualitative methods, including focus groups and interviews and development meetings to refine the intervention. So qualitative methods were really used throughout the whole process to develop the intervention. So moving on now to talk about how qualitative methods can be used sort of during and after trials. So qualitative methods can help evaluate the process and outcomes. A process evaluation can help understand how or why an intervention has worked. It can help understand how an intervention and an implementation strategy functions, and often a logic model might be developed to explain what an intervention sets out to do, and it can then be used to look at whether it actually achieved what it set out to do. Process evaluation can also tell us whether an intervention and an implementation strategy was followed as intended, so it can tell us a bit more about fidelity. And this is important whether the trial is a trial with a complex intervention or a CTIMP, or, a range of trials. So a good example of a process evaluation we've been involved in recently is from the EPATS study, which is led, I believe, by Professor Hastings and Dr. Gore, and the qualitative and the process evaluation was worked on by Jeremy Segrot. This process evaluation was embedded within EPATS, which was a feasibility trial of a group parenting programme for parents of children with intellectual disability. So the EPATS process evaluation used a mixed methods approach. So they used qualitative interviews with both parents and practitioners to look at barriers and facilitators to recruitment and implementation, to look at the impact on participants, but also to help decide which outcomes could be used in a future trial. So the qualitative data was collected, oops, sorry, zoomed on a bit too fast. The qualitative data was, trying to go back, never mind. The qualitative data was combined with quantitative data, which was collected on recruitment rates and implementation fidelity and the feasibility of outcome measures. So EPATS is a good example of a mixed methods process evaluation. Okay, and then finally, moving on to look at how we can evaluate outcomes and use qualitative research methods to do this. So the PUMA study is a recent study and is an excellent example of how we can evaluate outcomes. It's a program which was developed to improve systems in hospitals for detecting and responding to deterioration in child patients. So Amy Lloyd and team worked on this with Professor Davina Allen in the School of Healthcare Sciences. And this was a really large ethnographic, it involved a large ethnographic case study approach and an embedded process evaluation with over 400 hours of observations of clinical practice, over 100 staff interviews, and over 60 interviews with parents at 2 time points before and after the implementation of the PUMA program. So it really has led to an in-depth understanding of changes in practice, grounded in the local context for each site, which are really crucial to help understand the quantitative outcomes of the study. And I've done it again, gone on too soon. So one of the really important outcomes of the PUMA study was an implementation guide to identify the key ingredients to the successful implementation of the program. So again, a really good example of how qualitative methods can be used in a very meaningful way. So this has been a very quick overview of how we can embed qualitative research before, during and after trials within how we do it within the Centre for Trials Research. And I then particularly wanted to just focus on talking about an example of how members of the qualitative research group are using traditional qualitative methods in a different way. So Bethan Pell, who's a researcher in CTR, worked recently on the STAR family study, which was led by Dr. Rhiannon Phillips, who was working in the Division of Population Medicine at the time. And the STAR study explored the needs of women with autoimmune rheumatic disease throughout their journey to motherhood. And they wanted to use a qualitative method called the timeline method, which is used to encourage, it asks participants to form a visual chronological representation of a particular significant life event. So there's a template here on the slide, which shows an example of how one of the participants might begin their template. It's particularly good at empowering people to organise and share their own narratives in their own way, and it's particularly useful to encourage people to talk through sensitive but complex topics as well. So in this case, starting a family. The question was though, so this is a useful method to use, but the question was, can visual timelines still be used effectively in telephone interviews? Because this particular study wanted to carry out some telephone interviews with geographically diverse participants to make sure they reached a wide range of people. So this was the question to be asked, and Bethan Pell has written recently on this, and she found the quick answer is she found that yes, visual timeline method can be used within telephone interviews. They actually used they actually sent out a resource pack to participants before the interview with a timeline template, so the one I've just shown you here, but also some sort of creative items that people might use if they wanted to get creative. So they had coloured stickers and pens and things that they could really use to kind of take ownership of this timeline. The participants were given a set of instructions, and it was made clear that the template was just an example, and they could write as much or as little as they want, or even not write anything at all if they didn't want to. And it highlighted that the idea of this exercise was to help women tell their own story in whatever way they felt reflected their experiences. And in this particular instance, the timeline itself wasn't necessarily the data to be collected and analysed. It was more of a sort of way to facilitate telling a story, but the timeline method can be used, and the timelines can be collected and analysed in their own right as a form of data. So Bethan and colleagues found that yes, the timelines can be used.
To interrupt 1 second, I was wondering how many slides you got left, because we are
the last one. Sorry, yeah, last one.
Yeah.
So so they can. We found that they can be, or Bethan and colleagues found that the method can be used well. It can facilitate interviews. It can provide rich data. It can help the participants kind of retain ownership of their data. And we feel that this method is particularly timely, given that in COVID-19 times, so many researchers are moving to having to move to collect research data remotely. So we will continue to build an evidence base on how we can still generate these rich and detailed qualitative data whilst working remotely. So that's work in progress. And that is my last slide.
Thank you very much, Lucy. Just to let everyone know, obviously we've gone over a minute. It's now after 1 p.m. So I realise we have run out of time for the Q&A session, but all I can encourage you to do is write your questions in the Q&A box, and the speakers can respond to them before the next session. But otherwise we are back at 1:30 for the next session. Thank you for attending, and go and have some lunch, I think. Thank you, speakers.